Hello, and thank you for coming to this talk. My name is Adam Cooper. I'm a PhD student in atmospheric chemistry at the University of California, San Diego, where I research the atmospheric fate of organic UV filters as a part of the SLADE research group. Today I'll be presenting on some work I'm doing with Chemists Without Borders, with major contributions from Raquel Gilliland and Brandy Revels. We are a team made up of academic, nonprofit, and governmental professionals around the world who have joined forces to tackle a very specific problem championed by the GOES Foundation: synergistic toxicity of UV filters and microplastics on phytoplankton. At Chemists Without Borders, we realize that chemists are responsible both for building the molecules that makes our lives better and also their unintended impacts on the environment and human health. This project is built around organic UV filters found in sunscreens and used as plastic additives. These present ecotoxicological effects, most famously oxybenzone causing the bleaching of coral reefs, but also on marine life from algae to fish. They can biomagnify up the food chain, which presents another route of exposure to humans beyond their application as sunscreen agents. Oxybenzone specifically is tracked in my state of California in a biomonitoring program for its impact on endocrine disruption as a phenolic estrogen mimic. These compounds also show genotoxic and carcinogenic effects. So why am I at Micro 2020 and not the sunscreen symposium? The unfortunate reality is that microplastics can compound this problem, as they do with many others in this environmental sphere. There's been increased attention on how microplastics can act as sorbent particulate for the accumulation and transport of persistent organic pollutants. And while organic UV filters are not officially POPs, they exhibit many of the same characteristics: long-lived, toxic, and hydrophobic compounds. This hydrophobicity means they will preferentially accumulate on the surface of hydrophobic microplastic particles. Thus, the same issues that arise from marine life, mistaking microplastics for food, also leads to an inoculation of these hydrophobic compounds on their surfaces. Microplastics are a vector for the accumulation, transport, and biological uptake of these chemicals. In the study here by Knott et al., they observed this synergistic toxicity of microplastic fragments and BP3, or oxybenzone, additives on a grazing zooplankton. They found having both compounds present led to an effective toxic dose nearly an order of magnitude more dilute than when either was present alone. This synergistic toxicity is being observed in many macroscopic creatures, but has yet to be characterized on the base of the food chain, phytoplankton. Microplastics can absorb onto the surface of these plankton, and nanoplastics may pass directly through their cell walls, potentially causing similar synergistic effects others have seen. This is still an unexplored field, and one we are pursuing with researchers down the hill from me at Scripps Institution of Oceanography. We welcome and hope others in the field will conduct these tests with a wide range of parameters and model systems so we can build a better understanding of this emerging threat. So why is this important in the Anthropocene? Phytoplankton are a tremendously useful ecological group that sequesters carbon dioxide from the atmosphere and produces the majority of the world's oxygen. This serves as a buffer for rising CO2 levels during the climate crisis, and the strength of this system has saved us from experiencing the full warming potential of our emissions. However, these phytoplankton populations have been decreasing at an alarming rate of 1% per year, and we have already seen marine net productivity halve since 1950. While most scientists attribute dying populations to a changing climate, leading to ocean acidification and stratification, the growing polluted chemosphere in the ocean cannot be helping. Combining the levels we see in environmental samples with toxicities we observe in experimental studies, there is a major emerging issue that does not seem to be slowing down. Losing the health of the oceans means losing the fight against climate change. If we can save the plankton, it brings us one step closer to saving the planet. The future looks bleak, yet all hope is not lost. One glimpse of positivity that has emerged from the COVID-19 pandemic has been global observations of the rebounding marine life in places most impacted by UV filter pollution. Tropical areas with large tourism industries that saw fewer travelers and beachgoers. In general, sewage and wastewater effluent have decreased, along with noise and air pollution. However, the production and waste of single-use PPE and other packaging material has increased the plastic waste stream. We are beginning to use volunteer-driven, top-down methods to observe what impact this may have on the world's oceans globally. To accomplish this, we are exploring NASA's MODIS chlorophyll A dataset, which serves as a decent proxy to phytoplankton populations and productivity. Our volunteer team has been hard at work to trace different tracks of coastal regions and islands to observe any deviation in natural variation of chlorophyll A concentrations during the COVID-19 pandemic. So far, they have been focusing on South America, the Arctic, and Hawaii. However, this data is inherently messy and may require longer timescales up to the end of this year to observe any perturbations caused by the lockdowns. We are a growing team and we thrive on collaboration with the experts such as you all, which is why we are here at Micro 2020 ringing our alarms. As our President Ray likes to say, we have a platoon of volunteers. We need an army. If you'd like to join our army, please reach out through my email listed below. Thank you, and I look forward to chatting with you during the live session.